A dark shoreline can suddenly ignite blue when breaking waves disturb millions of microscopic light-producing cells near. Only moving water flashes brightly, revealing that mechanical disturbance rather than moonlight creates the visible glow inside cells.
Footsteps compress and shear the water, triggering countless individual flashes that merge into one luminous trail across blooms. Each flash lasts briefly because a single cell releases only a limited packet of chemical light within seawater.
The apparent glowing wave is actually a crowd of microscopic organisms responding one cell at a time, rapidly.
Many luminous coastal organisms are dinoflagellates, single-celled eukaryotes propelled by two differently oriented flagella. Plankton is a community, not one organism, and only some members possess bioluminescent chemistry across glowing coastal waters.
Bioluminescence evolved in selected lineages, so neighboring plankton can experience the same force without flashing, inside active blooms.
Currents, tides, and shoreline geometry.
A dense population makes weak cellular flashes visible across large moving objects and entire wavefronts after disturbance. What appears continuous from shore becomes a field of discrete flashes when viewed at smaller scales, within seawater. Different disturbances create different shear patterns, shaping the brightness and geometry of the luminous response during rapid stimulation. Breaking waves combine turbulence, compression, and rapid deformation, producing strong stimulation across many suspended cells within living cells. The luminous sheet follows moving turbulence, then disappears once cells stop experiencing sufficient mechanical stress under moving waves. Nighttime darkness helps observers see bioluminescence, but darkness alone cannot create it. Coastal displays change rapidly because plankton abundance and water movement vary through time. This response links oceanic light. Several dinoflagellate species can create similar blue flashes while differing in size, ecology, and toxicity under moving waves. A glowing bloom is not automatically poisonous, and a toxic bloom is not automatically Understanding the glowing ocean requires following one mechanical signal from the cell surface to a photon during darkness. Mechanical force deforms the cell surface, beginning a rapid signaling pathway before any visible light appears after disturbance. Mechanosensitive structures convert physical stress into electrical activity across the dinoflagellate membrane. This mechanism clearly clarifies coastal dynamics. Internal ion changes help connect membrane deformation with the organelles responsible for producing light across glowing coastal waters.
Dinoflagellate cells contain a large vacuole whose proton gradient stores electrochemical energy for the flash across coastal waters.
Voltage-gated proton channels open during stimulation and allow hydrogen ions to move rapidly toward light-producing compartments. The proton movement acidifies specialized organelles called scintillones positioned close to the cell surface during local coastal observation. Each scintillon contains the molecular machinery needed for an ultra-fast, localized chemical reaction. This pathway controls cellular behavior. Luciferin supplies the light-emitting substrate, while luciferase catalyzes its oxidation when conditions become acidic across coastal waters. Acidification changes molecular interactions, freeing luciferin and activating luciferase within the same tiny compartment throughout nighttime marine observation. Luciferase combines luciferin with oxygen, converting stored chemical energy into an electronically excited product across glowing coastal waters. A photon appears when the reaction product relaxes from its excited state. This response clearly controls experimental light. Coordinated activation of numerous scintillones makes one microscopic cell bright enough to detect. This contrast clarifies ecological light. The complete response unfolds quickly because ion movement and enzymatic chemistry occur within tightly organized cellular structures, locally. Dinoflagellate bioluminescence is among the fastest visible whole-cell responses to mechanical stimulation. This defense clarifies mechanical dynamics.
Repeated stimulation can temporarily reduce available light capacity until cellular components recover.
Recovery restores proton gradients and reorganizes the molecular system for later flashes. This defense clearly reveals cellular behavior. Dinoflagellates integrate mechanical information rather than responding identically to every movement. This process clearly links coastal research and underwater. Cell size, wall structure, and membrane mechanics influence sensitivity to water motion. This defense clearly maps dynamic light.
Mechanical stress becomes visible light through a rapid mechano-electrochemical signaling chain. This pattern tracks nightly turbulence.
Bioluminescent capacity follows an internal circadian rhythm rather than remaining equally strong throughout every hour, under moving waves. Many photosynthetic species prioritize daytime carbon fixation and prepare their light system for the coming night, near shorelines. Cellular organization changes before darkness, positioning light-producing structures for efficient nighttime flashing. This defense changes mechanical patterns.
Biochemical readiness peaks during the biological night, even when cells remain under constant laboratory conditions, inside active blooms.
Persistent rhythms demonstrate that timing originates inside the cell, rather than responding only to immediate darkness, near shorelines. External light can reset or disrupt the cellular clock, controlling luminescent capacity. This pathway clearly tracks ecological responses. Sudden darkness cannot instantly replace the molecular preparation normally scheduled by the circadian system during controlled cellular stimulation. Dinoflagellate lineages can regulate light using different patterns of protein synthesis, destruction, and cellular reorganization under moving waves. Daily protein turnover creates a strong rhythm by repeatedly removing and replacing light-producing enzymes during rapid stimulation. Similar visible rhythms can emerge through different molecular controls. This pathway clearly clarifies marine ecology during nighttime disturbance.
Organelle position may change across the daily cycle, altering how efficiently stimulation triggers light during controlled cellular stimulation.
Circadian timing ensures defensive light is strongest when darkness makes photons most visible. This method records mechanical motion. Emitting light under bright sunlight would provide less visual benefit while consuming limited chemical resources inside active blooms.
Nighttime light may be especially useful when grazers feed under low visibility conditions. This mechanism reveals nightly research. Population rhythms can synchronize broad coastal displays across millions of independent organisms. This cycle clearly connects mechanical hazard. Nighttime capacity can transform ordinary turbulence into visible shoreline luminescence. This rhythm clearly supports mechanical dynamics underwater. Weather affects what observers see, while the cellular clock determines what organisms can produce inside dense bloom waters. Coastal light pollution may alter biological timing in plankton exposed near illuminated shorelines. This rhythm records nightly signaling. Daily timing separates energy capture from the period when defensive light is most effective throughout nighttime marine observation. The glowing ocean depends on both mechanical disturbance and a cell prepared by its internal clock under stimulation. Heating currents and direct contact mechanically stimulate cells near an approaching grazer. This pathway clearly controls oceanic light. A sudden photon burst can startle small predators and interrupt their feeding behavior. This rhythm supports ecological hazard. Laboratory experiments show that bioluminescent flashes can reduce grazing success in some predator-prey interactions within living cells. Defensive value becomes clearer when luminous and non-luminous prey experience identical predators. This contrast clearly supports nightly signaling. Light may also expose a small grazer to larger visual predators nearby. This system clearly maps mechanical hazard. This proposed burglar alarm effect recruits a predator that threatens the Dinoflagellates attacker. This pattern tracks nightly research. A secondary predator can indirectly protect luminous cells by removing their immediate consumer. This response reveals local hazard. Different predator communities may favor different benefits from the same flash. This cycle clearly clarifies coastal behavior underwater. In some species, light might reinforce warning signals associated with toxicity or poor food quality across coastal waters. Aposematic interpretations remain less established and probably vary among species and predators. This response clearly changes visible responses. A moving predator can leave a luminous trail that reveals its route through plankton throughout nighttime marine observation. Larger swimmers generate broad mechanical disturbances, producing glowing outlines without necessarily being harmed. Bioluminescence can trace animal movement across darkness, making invisible hydrodynamic patterns suddenly visible. Human movement can stimulate the same cellular defense pathway on a much larger physical scale, under moving waves. Each cell responds locally, so the glowing pattern mirrors where mechanical stress travels. This rhythm connects experimental motion.
Even small impacts can trigger light when cell density and nighttime readiness are sufficiently high. During rapid stimulation, wave glow emerges from countless defensive responses accidentally triggered by non-living turbulence. This process clearly reveals nightly behavior. The cellular mechanism responds to both biological threats and ordinary physical motion. This defense clearly tracks mechanical hazard.
No single experiment proves that every luminous species evolved light for the same ecological reason, under moving waves.
A spectacular beach display may be the unintended landscape-scale result of microscopic survival strategies across coastal waters. Visible bioluminescence usually required enough luminous cells to accumulate within the water column. This method clarifies dynamic responses. Physical transport can create intense local displays without increasing total regional cell numbers. This rhythm reveals visible motion. Nutrients can encourage blooms, although species respond differently to temperature, salinity, mixing, and food availability. Inside active blooms, some luminous dinoflagellate blooms look discolored by day and brilliant when mechanically disturbed at night. Across coastal waters, daytime color is not a reliable test for nighttime bioluminescence. This cycle clearly clarifies cellular behavior during darkness. Light production and toxin production are separate biological traits. This pattern clearly supports nightly behavior during nighttime disturbance. Some species possess both traits, making identification more important than visual appearance. This contrast clearly supports marine signaling. Certain harmful dinoflagellates transfer toxins through food webs, even when the water looks ordinary, inside dense bloom waters. Large blooms can also cause ecological stress when decomposition consumes oxygen. This signal clearly links ecological behavior underwater. Oxygen depletion often results from microbial decomposition rather than the visible glow itself. This process explains coastal patterns. Observers cannot determine safety by brightness, color, or smell alone. This pathway clearly maps mechanical light during darkness. Species identification and environmental measurements provide stronger evidence than appearance. This contrast clearly reveals marine light during darkness. Environmental monitoring helps explain why a bloom formed and whether conditions are deteriorating. This system records oceanic dynamics.
Remote sensing which tracks large blooms while nighttime bioluminescence still requires specialized observation.
Forecasting luminous events depends on biological abundance and physical transport. This system clearly records cellular responses during darkness.
Mixing can rapidly dilute cells and change visibility. This signal clearly records local readiness during nighttime coastal disturbance. The same regional population may produce strong glow only where local circulation gathers it, throughout nighttime marine observation. These properties must be evaluated independently rather than assumed from one glowing observation. This system tracks cellular turbulence. The organisms may remain present even when their flashes are too sparse for human eyes, under moving waves. Scientists quantify mechanosensitivity by applying known forces while recording the timing and brightness of individual flashes near shorelines.
Mechanical models connect cell wall properties with the force required to trigger signaling. This cycle clarifies dynamic research.
Channel experiments test which ion movements are necessary for light activation. This pattern clearly connects dynamic ecology underwater. Population measurements estimate bioluminescent capacity across species, seasons, and environmental treatments.
Microscopy reveals how different regions of one cell activate during stimulation. This system clearly maps visible dynamics underwater.
Researchers build living materials that convert mechanical deformation into visible biological light. This response clearly explains marine research.
Biohybrid systems demonstrate that mechanosensitive luminescence can function inside engineered soft materials. Living sensors can visualize fluid pressure without batteries. This process clearly clarifies cellular turbulence during nighttime coastal disturbance. Engineered applications require light cycles, nutrients, seawater chemistry, and living cell maintenance. This mechanism clearly changes ecological behavior. Dinoflagellate light responses can reveal environmental stress affecting cellular function. This rhythm clearly tracks nightly light during darkness. Bioassays compare biological effects rather than identifying every contaminant directly. This pathway clearly tracks visible readiness during darkness. Bioluminescent plankton can visualize flow patterns that are difficult to observe otherwise. This pathway clearly clarifies biological hazard. Each luminous path records where suspended cells experienced sufficient shear. This pattern clearly connects local signaling during darkness. Living tracers provide qualitative and quantitative information about fluid mechanics by illuminating shear, vortices, and deformation around structures. Field studies connect laboratory mechanisms with complex ocean conditions. This contrast clearly links mechanical signaling. Vertical migration shifts luminous populations through the water column, changing when surface waves encounter the highest cell densities. Similar blue light can serve different ecological functions and arise from different organisms. This mechanism separates experimental signaling.
The glowing shoreline begins with one cell sensing force and converting chemical energy into a photon across blooms. The ocean appears to glow because millions of microscopic defensive flashes synchronize with the motion of water nightly. One New Guinea songbird carries powerful poison through its skin and feathers. The hooded pitahui lives only within New Guinea's forested landscapes. Dense rainforest conceals a medium-sized bird with striking black and red plumage.
Its contrasting colors make the species immediately recognizable.
Bold coloration may help advertise an unpleasant or dangerous defense.
Potential predators could learn to avoid the bird after negative encounters.
Scientists discovered the toxin while conducting ordinary bird research.
Handling the bird produced unexpected burning and numb sensations. Those sensations suggested a biologically active chemical on the plumage.
Field observations prompted chemical testing of feathers and tissues.
Researchers compared toxin levels across different body tissues.
Chemical separation isolated a potent steroidal alkaloid.
The compound belonged to the batrachotoxin family.
Previously, these toxins were famous from poisonous frogs across another continent. Unrelated animals had acquired remarkably similar chemical defenses. The similarity pointed toward environmental acquisition rather than shared ancestry.
Diet became the leading explanation for how the bird obtained poison. The original dietary source was not immediately known. Toxin levels varied among birds collected in different places. Not every individual carried the same chemical concentration.
Batrachotoxin targets voltage-gated sodium channels in nerves and muscles.
Sodium channels normally open briefly during electrical activity. Batrachotoxin stabilizes channels in an open state.
Continuous sodium entry disrupts the membrane's electrical balance. Affected cells cannot restore normal excitability.
Muscle control also depends on properly timed sodium currents.
Cardiac electrical activity can also be disrupted.
The toxin acts by changing channel behavior rather than blocking the pore. Its chemistry allows close interaction with channel proteins. Structural studies reveal multiple contacts between toxin and channel. Binding favors an abnormal open configuration.
Many natural toxins target these essential electrical gates.
Batrachotoxin is distinctive because it drives persistent activation.
A painful encounter can teach avoidance without requiring ingestion.
Learned avoidance could make small toxin quantities effective. Poison adds another protective layer to ordinary bird defenses. External tissues contain the highest measured toxin concentrations. Surface placement improves contact with attackers or parasites. The bird effectively wears a chemical defense. Toxic compounds occur directly within the plumage. Contact can deliver an immediate, unpleasant warning.
Researchers reported tingling, burning, and numbness after handling birds.
Those reports revealed toxicity before full chemical identification.
Skin and feathers consistently held the strongest concentrations. Internal distribution appears uneven rather than uniform. The bird must transport dietary toxin without poisoning itself. Toxin is ultimately deposited into external tissues. New plumage may incorporate compounds acquired from food.
Molting may change the bird's total toxin reservoir. Toxic plumage may also affect feather parasites.
Anti-parasite benefits remain plausible but incompletely measured.
Loose feathers could reinforce predator avoidance.
Toxic feathers might influence the nest environment.
Whether chicks receive meaningful protection remains uncertain.
Toxin accumulation may depend on diet after hatching.
Pittohus eat many arthropods rather than one exclusive prey.
Researchers searched stomach contents for a toxin-bearing source.
One little studied beetle group produced a compelling clue. The dietary trail pointed toward choricine beetles.
Choricine beetles contain high levels of batrachotoxin compounds.
Field samples confirmed that pittohus eat these beetles.
Diet offers a plausible route for toxin acquisition.
The compound must escape normal metabolic destruction. Unknown transport mechanisms likely move toxin into the body. Selective distribution suggests regulated sequestration. Geographic variation may reflect differences in available prey. Dietary exposure could explain variable toxin loads. Seasonal prey changes may alter chemical defense. Direct seasonal tracking remains limited.
kerosene may not be the only source.
The ultimate producer of batrachotoxin remains unknown.
Scientists have not proven where the food web first creates the molecule.
Chemical presence does not demonstrate biosynthesis.
Tracing labeled precursors could test where synthesis occurs.
Metabolic tracing could distinguish production from sequestration.
Structural continuity would support direct dietary transfer. Chemical fingerprints may connect predator and prey. Shared compounds strengthen the food chain hypothesis. Acquiring poison creates a second problem: avoiding self-intoxication.
The bird's own sodium channels should be vulnerable to batrachotoxin. A simple target site mutation once seemed like the obvious defense.
Recent evidence does not support an easy mutation-based explanation.
Resistance may instead involve several protective mechanisms.
Carrier proteins could reduce exposure of sensitive tissues.
Rapid sequestration may keep circulating concentrations low.
Compartmentalization could isolate toxin from vulnerable channels.
Tissue distribution may provide part of the protection. Metabolic transformation may also contribute, but evidence remains sparse. Any detoxification system must preserve useful poison in external tissues.
Self-protection and toxin storage must operate together.
Population comparisons could reveal resistance-related genes.
Genomic studies can identify candidate protective pathways. Resistance may be distributed across multiple systems.
Functional tests are needed to confirm protection.
Candidate mechanisms must change measurable toxin sensitivity. Poison frogs face the same self-resistance challenge. Similar chemical defenses need not use identical protection.
The bird's resistance mechanism remains a major unsolved problem.
The complete pathway from prey to plumage is still incomplete. Chemical defense can reduce the value of attacking the bird.
Taste or contact may create rapid avoidance.
Predators can associate plumage with an unpleasant experience.
Similar coloration raises questions about warning signals and mimicry. Color alone cannot establish mimicry.
New Guinea communities recognized these birds as unpleasant long before laboratory discovery. Traditional knowledge preserved practical information about toxicity.
Scientific understanding grew by connecting local knowledge with chemical analysis.
Archived specimens can reveal changes across decades.
Modern methods can measure toxin while preserving valuable material.
Historical samples could track geographic and temporal variation.
Environmental shifts may alter access to toxin-bearing prey.
Chemical defense may weaken if the dietary source disappears. Protecting the bird also means protecting unseen ecological relationships.
One defensive molecule can connect several trophic levels.
Two crucial links remain unresolved.
Scientists still do not know where the molecule is first synthesized.
They also lack a complete resistance mechanism.
The established facts make the remaining mystery even more compelling.
Field evidence shows that the hooded pitohui turns an unknown dietary toxin into one of nature's strangest defenses. This Antarctic fish reaches adulthood without red blood cells, leaving its circulating blood pale and nearly transparent. Its entire evolutionary story unfolded within the isolated, freezing waters surrounding Antarctica. The Antarctic circumpolar current helped isolate southern fishes from warmer oceans for millions of years, supporting southern ocean separation. These seas remain cold enough to freeze the body fluids of most unprotected fishes, central to sub-zero seawater exposure. Antifreeze glycoproteins prevent dangerous ice crystals from growing inside many Antarctic nodovoid fishes, central to antifreeze crystal control. These proteins attach to ice and interrupt the repeating structure needed for further growth, supporting ice-binding molecular defense. Cold water slows biochemical reactions, movement, digestion, and nearly every other process inside the animal, supporting cold-slowed biochemistry. A comparatively low metabolic demand reduces the amount of oxygen its tissues must receive each minute, supporting reduced metabolic demand. Nearly every other vertebrate transports oxygen using red cells packed with hemoglobin, while preserving standard vertebrate oxygen transport. Hemoglobin multiplies blood's oxygen capacity by reversibly binding gas molecules inside circulating cells, which affects hemoglobin oxygen binding. Icefish blood lacks both the red cells and their dominant respiratory pigment, sustaining translucent plasma circulation throughout Antarctic activity. Under a microscope, its circulation appears strikingly different from ordinary vertebrate blood, which affects erythrocyte-free blood samples. Oxygen must travel physically dissolved within plasma rather than attached to hemoglobin, sustaining plasma-dissolved oxygen throughout Antarctic activity. That method carries only a small fraction of the oxygen transported by red-blooded relatives, supporting weak oxygen delivery. Losing hemoglobin reduced blood oxygen capacity by roughly an order of magnitude, with consequences for collapsed carrying capacity. Yet the fish survives because its entire circulatory system compensates for that enormous deficit, supporting whole system compensation. No single adaptation explains life without red blood. Several mechanisms work together continuously, which cold seawater naturally holds more dissolved oxygen than warm seawater, compensated by cold water oxygen abundance inside Antarctic tissues. Abundant environmental oxygen improves diffusion across the gills, despite weak transport inside the blood, which affects gill diffusion efficiency. The Antarctic environment made a normally catastrophic blood defect temporarily survivable. Adult crocodile icefishes are the only known vertebrates completely lacking circulating red blood cells, central to adult erythrocyte absence. Their developmental program no longer produces the mature oxygen-carrying cells typical of vertebrates, supporting arrested blood cell development. Genomic evidence records the breakdown of genes required for functional hemoglobin, compensated by globin gene degeneration inside Antarctic tissues. Key globin genes were deleted or reduced to non-functional genomic remnants, compensated by non-functional genomic remnants inside Antarctic tissues. Globin loss occurred after the icefish lineage separated from hemoglobin-producing notothenoids, while preserving post-divergence hemoglobin loss. Modern white-blooded icefishes inherited the condition from a common ancestor, compensated by shared ancestral deletion inside Antarctic tissues. The family later diversified while retaining its unusual blood physiology, compensated by icefish family diversification inside Antarctic tissues. Myoglobin followed a different evolutionary path and disappeared independently in several icefish species, which affects independent myoglobin loss. Where present, myoglobin helps buffer oxygen inside constantly active heart cells, revealing cardiac oxygen buffering under stable cold conditions. Some icefish hearts function without either hemoglobin or cardiac myoglobin, sustaining myoglobin-free heart function throughout Antarctic activity. Independent losses demonstrate that similar physiology evolved repeatedly within the family, compensated by repeated evolutionary loss inside Antarctic tissues.
Scientists still debate whether hemoglobin loss was advantageous or merely survivable under extreme cold.
Removing red cells made blood easier to pump while severely weakening oxygen transport, which affects viscosity-capacity trade-offs. Lower viscosity reduces resistance within the circulation, partly offset through low-resistance plasma flow across cold tissues. Greater cardiac output partly offset the small amount of oxygen carried per unit blood, which affects elevated cardiac output. Wide vessels allow high-volume flow without excessive pressure, partly offset through wide vessel circulation across cold tissues. Dense capillary networks shorten the distance between plasma oxygen and tissue cells, tied closely to dense capillary architecture. Mitochondrial abundance increases the cellular surface available for oxygen use, partly offset through mitochondrial oxygen use across cold tissues. Icefish mitochondria also display unusual density, size, and internal organization, partly offset through unusual mitochondrial morphology across cold tissues.
Hemoglobin loss reshaped anatomy from genes to vessels, heart, and cellular energy machinery, especially through genome-to-heart compensation.
Every delivered oxygen molecule begins as gas dissolved within the surrounding seawater, while preserving gill plasma oxygen transfer. Lower temperature increases the amount of oxygen water can hold, compensated by temperature-dependent oxygen solubility inside Antarctic tissues. Polar conditions therefore maximize the oxygen available for passive uptake, compensated by polar passive uptake inside Antarctic tissues. Thin exchange surfaces reduce the distance oxygen must diffuse, partly offset through thin membrane diffusion across cold tissues. No cellular carrier captures the gas after it enters circulation, revealing carrier-free circulation under stable cold conditions.
Carrying more plasma increases the total dissolved oxygen moving through the animal, sustaining expanded blood volume throughout Antarctic activity. Icefishes maintain substantially greater blood volume than many related fishes, partly offset through high plasma volume across cold tissues.
Cell-free blood flows with comparatively low resistance, partly offset through low cellular crowding across cold tissues.
High vascular density increases the area available for oxygen delivery, compensated by vascular oxygen delivery area inside Antarctic tissues. Short diffusion distances compensate partly for low oxygen concentration, partly offset through short tissue diffusion across cold tissues. Delivery succeeds through broad distribution rather than concentrated oxygen cargo, partly offset through distributed capillary delivery across cold tissues. Behavior also keeps oxygen demand within the circulation's restricted capacity, partly offset through behavioral oxygen conservation across cold tissues. Intense activity can challenge a system with little oxygen reserve, partly offset through burst swimming limitation across cold tissues. Icefishes generally possess limited scope for prolonged high-performance swimming, partly offset through low aerobic scope across cold tissues. Warmer conditions raise metabolic demand while lowering water's oxygen content, compensated by warming-driven oxygen mismatch inside Antarctic tissues. Temperature therefore compresses the animal's physiological safety margin from both directions, compensated by shrinking physiological margin inside Antarctic tissues. Its unusual blood works best within the environment where it evolved, compensated by stable sub-zero performance inside Antarctic tissues.
Depth and local circulation can provide relatively stable thermal conditions, compensated by deep water thermal refuge inside Antarctic tissues.
Shallow populations may encounter faster environmental change through shallow water exposure, limiting stress while sustaining water exposure.
Environmental oxygen and cardiovascular compensation remain inseparable parts of the same survival system, which affects environment circulation coupling. Icefish hearts are enlarged relative to those of similarly sized red-blooded relatives, which affects enlarged ventricular anatomy. Each beat moves an unusually large volume through the circulation, supporting high stroke volume throughout Antarctic cardiac activity. High stroke volume raises cardiac output without requiring extremely rapid heart rates, with consequences for cardiac output compensation. Cold cardiac muscle contracts slowly and depends on specialized cellular machinery, compensated by cold cardiac machinery inside Antarctic tissues. Mitochondrial density supports aerobic energy production inside the oversized heart, partly offset through heart mitochondrial density across cold tissues. Dense vessels keep oxygen pathways short within cardiac tissue, partly offset through short cardiac diffusion across cold tissues. Some species add myoglobin storage within the heart, partly offset through retained cardiac myoglobin across cold tissues. Other species survive after losing cardiac myoglobin independently, partly offset through independent myoglobin absence across cold tissues. Different lineages reach similar physiological outcomes through distinct genetic changes, partly offset through convergent cardiovascular outcomes across cold tissues.
Elevated nitric oxide signaling may help promote wide vessels and vascular growth, tied closely to nitric oxide signaling.
Nitric oxide regulates both blood flow and formation of new vessels, compensated by angiogenic vessel growth inside Antarctic tissues.
Long-term signaling can reshape anatomy throughout growth, through developmental compensation.
Vascular differences emerge as the animal develops, through heart growth trajectories, limiting stress while sustaining heart growth trajectories in Antarctica.
Static signals may transform a blood defect into a stable body plan, sustaining homeostatic circulatory remodeling throughout Antarctic activity. Low viscosity makes large volume pumping mechanically easier, partly offset through viscosity-enabled high flow across cold tissues. Tips. Cardiovascular compensation still requires substantial energy. Through energetic compensation costs, limiting
the fish pays for weak blood with a larger circulatory apparatus, revealing large circulatory investment under stable cold conditions. Warmer water can push that enlarged system toward its performance ceiling, sustaining thermal cardiac limits throughout Antarctic activity. Cardiac capacity may limit acute warming tolerance in some species, partly offset through cardiac performance ceiling across cold tissues.
Every compensation traces back to an ancient loss recorded in the genome, with consequences for integrated polar physiology. Their closest red-blooded relatives still retain that ancestral system, partly offset through retained relative physiology across cold tissues.
A major deletion destroyed essential hemoglobin coating material, partly offset through beta-globin deletion across cold tissues. Additional remnants preserve a molecular fossil of the lost pathway, compensated by alpha-globin genomic fossil inside Antarctic tissues.
Once the protein was no longer required, disabling mutations accumulated more freely, central to post-loss mutation accumulation.
Blood physiology changed far beyond the globin genes themselves, partly offset through reduced iron physiology across cold tissues.
White blood contains dramatically less iron bound in respiratory pigment, compensated by iron-poor white blood inside Antarctic tissues. The absence of red cells lowers viscosity substantially, partly offset through erythrocyte-free viscosity across cold tissues.
Lower viscosity may have softened the damage without making hemoglobin loss beneficial, sustaining evolutionary trade-off interpretation throughout Antarctic activity. Environmental conditions created a rare window in which the mutation could persist, sustaining cold ancestral tolerance throughout Antarctic activity.
Similar gene loss would be far less tolerable in warmer, oxygen-poor seas, which affects warm water incompatibility.
Red-blooded relatives often retain greater capacity for oxygen transport during thermal stress, which affects relative thermal capacity. Extreme specialization can increase success in stable environments while reducing resilience to rapid change, supporting specialization. Genome studies reveal additional adaptations beyond blood and circulation, partly offset through antioxidant gene expansion across cold tissues. Oxygen-rich water and abundant mitochondria can increase oxidative stress, partly offset through mitochondrial oxidative pressure across cold tissues. Strong antioxidant and repair systems limit damage from reactive molecules, partly offset through macromolecular repair defense across cold tissues. Icefish survival depends on a broader Antarctic molecular toolkit, partly offset through broader cold adaptation across cold tissues. Traits optimized for constant cold may become liabilities during warming, partly offset through warming-created mismatch across cold tissues.
Climate change modifies several ecological pressures simultaneously through multi-factor climate pressure.
Risk will vary among species, locations, depths, and life stages, partly offset through species-specific exposure across cold tissues. Warm water holds less dissolved oxygen while animal metabolism accelerates, partly offset through warming oxygen decline across cold tissues. Icefish face simultaneous reductions in supply and increases in demand, compensated by aerobic supply-demand compression inside Antarctic tissues.
Limited oxygen carrying capacity may bring some ice fishes to their ceilings sooner, tied closely to earlier performance ceilings. Thermal vulnerability is not identical across the family, partly offset through variable family vulnerability across cold tissues. Behavioral movement may reduce exposure to warming surface layers, partly offset through behavioral thermal refuge across cold tissues. Mutagens. Suitable cold refuges may not exist everywhere. Through geographic restraints, limiting stress, while
early life stages may encounter different thermal risks than adults, compensated by larval thermal exposure inside Antarctic tissues. Warming can reshape development before circulatory compensation is fully established, balanced through developmental oxygen stress during polar life. Changing prey fields can indirectly affect icefish energy budgets, partly offset through changing prey networks across cold tissues. Reduced food could make costly cardiovascular compensation harder to maintain, partly offset through cardiovascular energy deficit across cold tissues. Field monitoring is essential for separating short experiments from population trends, with consequences for long-term population monitoring. Research collections reveal anatomy, genetics, diet, and physiological condition, partly offset through research specimen collection across cold tissues. Modern tools connect whole animal performance with molecular mechanisms, partly offset through integrated molecular analysis across cold tissues. Comparative studies identify which traits followed hemoglobin loss and which evolved earlier, with consequences for comparative evolutionary mapping. Variation within the family turns icefishes into a natural evolutionary experiment, with consequences for within-family physiological variation.
Biology offers insights into oxygen transport without providing a direct model for human treatment, supporting biomedical research boundaries.
Genes. Understanding compensation may illuminate general principles of cardiovascular plasticity, partly offset.
Those adaptations remain inseparable from the cold, oxygen-rich sea that made them possible, helping explain Southern Ocean dependence.
Its future depends on whether physiology can keep pace with changing Antarctic water, tied closely to future warming pressure.
Antarctic icefish survive without red blood by rebuilding nearly every other part of oxygen delivery, supporting integrated oxygen delivery.
This South American fish can activate another animal's muscles without touching it, using electricity generated inside its own body.
Electric eels inhabit slow rivers, floodplains, and seasonally changing waters across northern South America, especially within quiet backwaters.
The habitats are dark, cluttered, and difficult to search visually, making electrical sensing far more useful than eyesight.
Electric sensing turns murky water into a readable electrical landscape. The advantage matters during dark water navigation.
Despite its common name, an electric eel is a South American knife fish rather than a true eel.
A long anal fin provides smooth forward, backward, and hovering movement, while the elongated body remains nearly straight. Precise hovering keeps the sensory field aimed steadily, while the animal investigates prey hidden among roots and leaf litter.
Electric eels regularly reach the surface because atmospheric air supplies most of the oxygen supporting their metabolism.
A highly vascular mouth lining absorbs oxygen after every surface gulp. Control strengthens during fast predator escape. Air breathing permits survival in stagnant pools, where dissolved oxygen can fall below levels tolerated by many fishes. Most of the elongated body behind the vital organs contains specialized tissues devoted to generating electrical discharges.
Electrocytes are specialized cells arranged so their tiny voltages add together. That helps during muscle cell transformation.
Evolution transformed muscle-derived cells by reducing contraction, expanding membrane specialization, and coordinating their electrical activation together. Asymmetric nerve input lets each electrocyte create a directional voltage difference. Control strengthens during synchronized neural firing. A neural command opens ion channels almost simultaneously across the organ. Efficiency rises during sodium channel opening. Each activated cell contributes only a small electrical potential. Thousands combine to create the organ's full voltage.
Series arrangement adds the tiny voltage from each cell, producing a much larger discharge across the complete organ.
Parallel arrangement increases available current, while series arrangement raises total voltage. That helps during parallel current amplification.
Surrounding water completes the external circuit whenever a high-voltage pulse travels from the eel toward another animal.
Genetic and anatomical evidence now separates electric eels into 3 living species, notably during species-level divergence. Each recognized species occupies a different combination of regional geology, water conductivity, and evolutionary history across Greater Amazonia. Electrical performance changes with the conductivity of surrounding water. Conductivity reshapes both current pathways and target exposure. Water chemistry reshapes the current pathway, changing how strongly a discharge crosses prey, predators, and surrounding habitat. Voltage alone cannot describe the biological effect of an electric discharge. Performance improves during multi-factor electrical effects.
Current, pulse duration, conductivity, and contact geometry together determine the biological effect of any electrical discharge underwater.
During a powerful pulse, the eel's head and tail behave as opposite electrical poles within the surrounding water.
Body position lets the animal shape where the strongest current travels. Precision increases during body position control.
Weaker pulses serve continuous sensing and communication, while powerful volleys are reserved mainly for predation and defense.
Objects with different conductivity distort the eel's self-generated field, allowing nearby structures to be detected without vision. Electroreceptors distributed across the skin sample tiny field changes created by nearby objects, prey, and environmental boundaries.
Electrical distortions become spatial information inside the nervous system. That neural map guides movement through cluttered darkness.
Active electrolocation reveals obstacles even when vision contributes almost nothing. The advantage matters during obstacle shape reconstruction. Living prey produces a distinctive electrical signature compared with stones, wood, sediment, and empty surrounding water nearby. Movement changes the pattern repeatedly, helping the eel separate prey from static background clutter during prey tracking. Pulse timing can change with context, arousal, and nearby electrical information. That helps during context-dependent pulsing. Weak electric discharges also carry social information between individuals without triggering a predatory or defensive attack underwater. Electrical signals can help distinguish species, individuals, and behavioral states. Performance improves markedly during species signal recognition. After detecting prey, the electrical system rapidly shifts from environmental mapping toward high-voltage muscular control instantly. A hunting eel can activate nearby prey motor neurons remotely, forcing muscles to respond before physical contact occurs. The prey's own nervous system carries the eel's externally imposed command. This matters during prey nerve firing. High-frequency volleys force simultaneous contraction across the prey's body, producing a brief period of whole-body muscular tetanus. A temporary tetanus blocks coordinated escape during the eel's final approach through cluttered, low-visibility habitat underwater.
Controlled experiments reproduce the same muscular response by applying eel-like electrical stimulation directly to isolated prey preparations.
Closely spaced pulses prevent full relaxation between twitches, merging separate contractions into one sustained, forceful muscular response.
Prey becomes immobilized because its own nerves and muscles obey externally imposed electrical stimulation from the eel.
Electricity does not physically push the target. Instead, it recruits the target's motor neurons and muscle fibers.
External voltage crosses the threshold needed to initiate action potentials. That becomes valuable during motor threshold crossing. Neural activation closes the timing gap between detection and capture. Control remains useful during rapid neural conduction.
A sudden high-voltage burst can interrupt an escape turn before the prey completes the movement or accelerates away. Immobilization creates a short capture window, allowing the eel to close distance before coordinated swimming can resume.
Curling positions both electrical poles near the target. This bioelectric mechanism remains important during opposite pole positioning.
This posture greatly increases current through the prey's body. Curling therefore amplifies stimulation without raising internal output.
Curling strengthens stimulation through geometry, increasing current across the prey without raising the organ's internal voltage further.
Repeated volleys keep struggling prey from regaining coordinated movement. Continued stimulation preserves the eel's brief capture advantage. High-voltage hunting carries a metabolic and electrical cost. Recovery between attacks restores ion gradients across electrocyte membranes.
Between attacks, membrane pumps rebuild sodium and potassium gradients across thousands of electrocytes inside the electric organs.
Recharge depends on active transport, resetting thousands of biological battery cells. This matters during ion pump resetting. When prey remains hidden, the eel emits brief electrical doublets that can force an involuntary muscle twitch from concealment. That twitch moves leaves, sediment, or water, revealing the hidden animal against an otherwise motionless underwater background.
Laboratory doublets reproduce the same rapid muscular twitch observed when eels searched naturally for concealed prey underwater. Spacing between two pulses is well suited to summating the prey's muscle response into one detectable movement.
After delivering a doublet, the predator pauses briefly and monitors the surrounding electric field for nearby motion. Electrolocation converts the involuntary twitch into a precise positional clue. This remains important during twitch signal detection. Multiple viewpoints improve localization inside cluttered cover. That electrical strategy provides major advantages during multi-angle localization. Movement distinguishes living prey from inert debris more reliably than static electrical shape within cluttered underwater habitat. Once the target is located, the eel immediately replaces probing doublets with an immobilizing high-voltage volley. The same bioelectric system first exposes concealed prey, then prevents that prey from escaping the approaching predator. A complete hunt combines weak electrolocation, forced twitching, high-voltage immobilization, body curling, and rapid physical capture. Low-voltage discharges continuously refresh the eel's environmental map before any predatory strike becomes necessary inside cluttered water. An initial field distortion suggests a possible living target, but cannot confirm identity without additional testing underwater. A doublet challenges the hidden object to respond biologically. Any twitch separates living prey from motionless environmental clutter. The resulting motion confirms that excitable tissue is present. The eel immediately converts confirmation into precise targeting. Twitch geometry helps aim the following high-voltage strike. A clearer orientation strengthens the following immobilizing volley.
The attack volley recruits the prey's own motor system, turning ordinary muscle physiology into a temporary restraint.
Electrical control reduces the need for a prolonged physical chase. This remains important during rapid target capture.
Bioelectric hunting becomes especially valuable where darkness, roots, and sediment make ordinary visual pursuits slow or unreliable.
After feeding, the animal returns to low-voltage sensing rather than maintaining energetically costly attack volleys afterward. Electric eels also direct powerful volleys toward large nearby threats that cannot be treated as ordinary prey. During shallow water defense, an eel may approach vertically and rise toward the intruder instead of retreating. Pressing the chin against a conductive target creates a more effective pathway for electrical current through the threat.
As the eel climbs higher from the water, measured current entering the contacted object rises progressively and dramatically.
Emerging from the water removes competing current pathways, concentrating more of the discharge through the contacted threat. Repeated high-voltage volleys during brief contact can persuade an approaching predator or intruder to withdraw rapidly. Alexander von Humboldt famously described electric eels rising against horses driven into shallow South American water during an early expedition. Modern experiments confirm defensive leaping, although they cannot verify every dramatic detail preserved in Humboldt's 19th century account.
Instrumented trials confirm that climbing contact increases electrical delivery. Measured current rose as the eel climbed higher.
The same electric tissues power both predatory strikes and defensive leaps. That helps during shared organ deployment. Sachs organ contributes weaker pulses used primarily for electrolocation, orientation, and communication rather than forceful attack underwater. Proteomic comparisons reveal distinct molecular profiles across the main, Hunter's, and Sachs electric organs within specialized tissues. Differences in channels, pumps, and structural proteins help each electric organ meet distinct energetic and signaling demands. Electric organs originate from muscle lineage cells during development. That process repurposes muscle precursors into non-contractile specialized electrical cells. During development, muscle precursors lose ordinary contractile machinery and gain the membrane specialization needed for electrical output. Electric organs evolved independently multiple times among fishes. Distant lineages repeatedly modified muscle through similar genetic pathways. Distant electric fish lineages repeatedly reused related genetic pathways while independently transforming muscle into electricity-producing tissue. Engineers examine electrocyte stacking for ideas about soft ion-based power sources, sensors, and bio-inspired materials today.
Bio-inspired research does not mean a living eel functions like a practical battery for ordinary human technology. Volta's electric eels have been observed gathering in groups exceeding 100 individuals near a remote Amazonian lake.
Coordinated movement herds prey into a dense, shallow aggregation. Herding concentrates many targets within one strike zone. Most eels maintain the encircling formation, while a smaller subgroup advances toward the compressed prey school together.
Several hunters then discharge high-voltage volleys nearly simultaneously. Timing several discharges together broadens the affected prey area.
Synchronized volleys can expose, stun, and scatter many small fish concentrated inside the shallow prey aggregations simultaneously. Nearby group members exploit the brief feeding opportunity created after several hunters discharge electricity together during feeding. The sequence can repeat during a single hunting event. Repeated cycles increase feeding efficiency under exceptional conditions. Similar events recorded in different years suggest structured social predation rather than one accidental gathering over time. Local prey density may make cooperative hunting worthwhile. Dense seasonal shoals can reward brief temporary social coordination.
Cooperative hunting likely appears only where prey density, shallow water, and local geography make coordination worthwhile for multiple hunters.
Group hunting is documented in Volta's electric eel, but should not be generalized across every species, habitat, or population.
Researchers still do not know how individuals coordinate each strike. The advantage matters during group coordination mechanisms.
Weak electrical pulses, body movements, and spatial position remain plausible signals for coordinating group attacks underwater together.
Field electrode arrays could separate individual discharge patterns during group hunts. This matters during field electrode monitoring.
Synchronized measurements may reveal whether attacks are actively coordinated. Combined data can expose timing of individual roles.
Protecting electric eel behavior requires conserving connected floodplains, tributaries, and seasonal channels throughout the greater Amazon basin.
Dams, pollution, and altered flood cycles can disrupt prey movements, breeding habitat, and seasonal hunting opportunities locally.
Healthy river connectivity preserves both electric eel diversity and the ecological conditions supporting their specialized behavior regionally. Together, electrolocation, remote muscle control, hidden prey detection, curling, leaping, and social hunting reveal exceptionally precise bioelectric behavior. Electric eels are living batteries that sense, command, defend, and sometimes hunt together through carefully controlled bioelectricity.
Some spiders leave the ground by releasing silk into the atmosphere.
Their journey can extend far